DNA contains nucleotides. Each nucleotide has three covalently bonded pairs. A nucleotide contains a nitrogen base, sugar, and phosphate. There are nucleotide bases: adenine, thymine, guanine, and cytosine.
There are three different types of DNA replication. There are semi-conservative replication, conservative replication, and dispersive replication. Semi-conservative replication produces two copies of DNA that each contain one of the original strands of DNA and a new strand. Conservative replication leaves the original DNA and produces two new strands of DNA. Dispersive replication produces two copies of the DNA where both copies contain an original strand of DNA and a new strand.
The helicase unwind DNA.
The RNA primase initiates DNA replication. RNA primase is an enzyme that forms short RNA segments called RNA primers. DNA polymerase attaches to each RNA primer. This is the beginning of elongation. DNA polymerase continues the new strand by adding new nucleotides in the 5 to 3 direction.
The leading strand is assembled continuously towards the replication fork. The leading strand and DNA polymerase moves in a 5 to 3 direction. Multiple RNA primer begin on the lagging strand, while DNA polymerase follow each primer. The lagging strand takes more time to.
The Okazaki fragments begin to form when DNA polymerase attaches to an RNA primer. These fragments contain short segments of complementary DNA. Another DNA polymerase replaces the RNA primers with DNA nucleotides.
The ligase enzyme joins the Okazaki fragment. It produces a single complementary strand of DNA.
The RNA primer is essential to DNA replication. It must be used to initiate each new fragment. The leading strand and every Okazaki fragment on the lagging strand begins with an RNA primer.